direct CEO of Bio Innovation Institute in Denmark. Basically, he received numerous awards, including Nature Mentor Award, Annie Award, Samsung Prime Minister Award, and then also he's a member of important academic institutes, including National Academy of Engineering in the U.S., Science in the U.S., Chinese Academy of Engineering, Royal Danish, and Royal Swedish. Also, he's a founding president of International Metabolic Engineering Society. So, without enumerating his award and honors, let me give him podium for today's lecture, Systems Biology of Human Metabolism. So, Jens, if you
thank you.
Yeah,
thank you very much. Yeah, thank you very much for the kind introduction, and thank you very much for again giving me this opportunity. And I'm very honored to give these lectures at KAIST. And as I said on Monday, I'm very sorry I cannot be there in physical presence. Hope to account for that in the future. But today I'm going to talk about the work that we have done on studying human metabolism. On Monday, I was talking about the work we've been doing on both systems biology and metabolic engineering of yeast. And I can say that some years back, we started to explore the opportunity of taking much of the modeling work we have been doing on yeast over several years and exploit that for going in and studying human metabolism. And today I will focus on this part of our research work in my research group. So there are a number of reasons for why you want to apply systems biology approaches to medicine or human health. First of all, one good argument is that what we have of current drugs are not really helping people they should. You can see this here with many of the top-selling drugs, that they, in worst cases, they are really treating 1 in 25 are actually really being helped by this. Besides this, there are a number of side effects, but maybe more importantly is that this actually hampers also development of new drugs, because in many cases it becomes simply so expensive to develop these drugs, and so we really need to be better in the future to cope with these things. And so there is quite some interest and developments also to go in this direction of personalized medicine, and what you can do, or what the objective here would be, that you basically follow individuals over time. And so instead of having a reference cohort, you can say when you do drug development, you actually use your own data for reference in terms of development of disease. And there is quite a lot of interest in that. Of course, if we are going to be able to do this in a large-scale fashion, meaning on a whole population basis, we of course need to have some measurement devices that allows us to get in and understand, for example, changes in physiology through measurement of for example, directly on your watch, or other types of external measurements. You can say in contact, non-contact measurements. But in order to get there, we will probably still need to get a lot of data on omics data. It can be on metabolomics, on plasma, and so on, in order to begin to understand, give this link between traditional phenotypes, specific biomarkers, and then link that with these type of non-contact measurements I was talking about. So really, that brings to the core of personalized medicine is really the identification of biomarkers, because it's first when we have good biomarkers, we can do this stratification and move from this traditional viewpoint here, where you have just a disease group and a control group, to you go to a situation where it's often much more complex. You may have people in many different segments, and this is particularly relevant for many of the large chronological diseases like diabetes, obesity, Alzheimer's, and so on, you often have people with many different comorbidities, and you need to be able to stratify such that you can better target the right medication to these people, as well as actually in the future develop the right medication for these individuals. And so why is it so hard to find biomarkers? Well, it is, of course, determined by a number of factors, what proteins and metabolites are floating around in our blood, or for that matter, what are present in our different tissues, because it is depending on the diet. The diet will directly impact our blood chemistry and what we take up of nutrients. And that will, of course, then deliver metabolites for metabolism in the different tissues in the human body, as well as they will secrete further metabolites back into the circulation system. And so therefore, when we draw a blood sample and going to analyze certain metabolites, it is a consequence on the past, you could say, the diet history, as well as the longer past in terms of how our tissues developed. But also, of course, by genetic factors and many others. So that's why we need to have a more directed approach to kind of search for such biomarkers, and I'll come back and talk about this later on. So really, metabolism is at the core of cellular function because it provides building blocks for all functions in the different cells and different tissues, provides free energy, it ensures the electron flow. But how do we really integrate all this information with new knowledge acquired, for example, on cancer cells? So how to do this? Here we use this concept that I was also talking about on Monday, and something we've developed extensively for yeast, but also for many other microbes, also in many other labs, including Sang Yoblib's lab in KAIST, developing these so-called genome-scale metabolic models. And these are comprehensive models that compiles information about all the stoichiometry of all the different reactions. So that, for example, the glucose goes into the cell, it gets phosphorylated by ATP. Next, it converts into fructose 6-phosphate, or so on. And we compile all the stoichiometry of all these reactions. We link this to specific enzymes. In some cases, we may have two different enzymes that are actually catalyzing the same reaction, so we call them isoenzymes. In some cases, we have one enzyme that is comprised of two different peptides, so it's a protein complex. But in all cases, we can begin to link the reactions with the specific genes in the genome. And this is quite important, therefore, of course, also to transcript as well as to proteins, because that determines, you could say, the different types of metabolism in the cell. Therefore, also, if we just go into the genome of the human body, or from a human, we will find that there are a number of genes encoding for enzymes. And that will represent, you could say, a generic model. This is the total metabolic capabilities of a human cell. But that does not necessarily reflect on which enzymes are expressed in a different cell type or different tissue, because that's only normally a subset of that. And I'll come back and talk a little bit about that. When we reconstructed this whole network here, we get to this metabolic network that we can also use for analysis, as well as also a mathematical representation that we can use for simulation. So, but one thing that is that we have really learned from this analysis and building these models is that these models are extremely complex, but they are also connecting metabolism maybe to a much higher extent than we had thought of before. And this is a graphical representation actually of yeast metabolism. Human metabolism will look exactly the same. You can see here that you have basically the specific metabolites. All the green dots are enzymes, but all the yellow ones are metabolites, for example, in the cytoplasm. And you can see how extensively connected it is. And that's because of using some key metabolites, but also cofactors like ATP, NADH, and NADPH. And then, of course, you can see that in certain subcompartments, there's a little bit more differentiated metabolism or separated metabolism, but still there is extensive exchange due to transport reactions and so on. So with this, With this graph, when you see this graph, this of course has clear implications as we're talking about on metabolic engineering on Monday, but it also tells us that any kind of perturbation that will happen can be a genetic factor. It can be because we get mutation due to environmental impacts. It can also be because we eat certain diets. There will be a perturbation in this network here, and this will spread out and actually give a metabolite, as we can call it, a distinct phenotype characterized by a certain set of metabolites that are in our blood. Now, this led me actually to propose a general hypothesis that I would argue that any disease that we will have will carry some sort of metabolic fingerprint in plasma or blood. And so when we talk about the use of GEMs, and particularly related for human metabolism, we can use them for several different things. We can compare metabolism between cells at the genome scale. We can identify co-regulated metabolic networks, so then we're using this network component. And we can also use these models for simulation of metabolic functions. And here we use the concept of flux balance analysis, where we balance metabolism around each specific node. And particularly this part here we can use for biomarker discovery. I'll give an example of that, whereas the simulations we can use for drug discovery. So we worked on these models for a number of years, but more recently we thought that we could actually begin to improve the predictive strength of these models by beginning to add information about enzyme kinetics and costs. And so what we do here is that we add, basically constrain the flux through individual reactions with an upper bound that is defined by kcat times the enzyme concentration. And this will give us a total constraint because the enzymes don't come for free, so to speak. So the total sum of all enzymes in the cell, of course, has an upper bound. And therefore, you cannot just allocate freely enzymes to all the different metabolic reactions in the network. And we developed this originally for yeast, and I'll come back and talk about that we also now expanded this to cover human metabolism. So let's focus on human genome scale metabolic models. So what we did some years back was that we developed a generic model. We collected information from multiple different places. We refer to this as Human Metabolic Reaction Database, HMR2. We collected this information. This original model had about 8,000 reactions linked to close to 4,000 genes and about 6,000 metabolites. And also we developed an algorithm that we refer to as T-init. It allows to look at which genes are expressed in different tissues. So it can also be if we have proteomics data. We use this information to then generate from this generic models for different tissues. It can be liver, it can be adipocytes, or it can be muscle tissues, or for that matter, for any kind of tissue. And the approach of this algorithm is basically that you say you have this network here, you run this init algorithm here, and then you say, okay, this enzyme is expressed in liver, this one is, and so therefore these reactions are in liver, this is present in liver and muscle, so therefore this whole pathway will be in liver, whereas the pathway is likely not going to be fully functional, for example, in muscle, because the first and the last enzymatic reactions are not there. So this is just an example of how this can be done. We've done this recently in collaboration with the Human Protein Atlas, collaboration with Professor Mathias Ullén from Royal Institute of Technology in Stockholm. And there they were doing RNAseq from 32 different tissue biopsies. And using those data, we then used these T-init algorithms to generate models for these 32 different tissues. Here we collected not the individual reactions, but the reactions collected into specific pathways. And here you can see, for example, that liver is the first one here. It basically has everything in human metabolism. There is one thing that falls out here, but really, generally, liver is almost like a representative of all the metabolic capabilities of the human body. Whereas if you go to other tissues, you can see they have many metabolic functions that they don't have. They rely either on delivering of some of these metabolites or degradation of them through the help of the liver. So we make all these models. They are available on our web portal, Metabolic Atlas. I'll come back and talk a little bit more about that also later. So one thing we thought first was to see, could we take this model here also and generate personalized models for, in this case, we wanted to look into to liver cancer, HCC, which is the most dominant form of liver cancer. And so we had proteomics data based on immunohistochemistry, again from Matthias Relain's research group, from 6 patients having HCC. And we compared that with data from the healthy cell types, as I was just showing before. And then what we did was that we looked into so-called antimetabolites. So these are metabolites that are basically inhibiting specific reactions. And when they do that, we then simulate if the cells can still function properly. If they cannot, that means that they will die, and therefore this is a potentially drug. But of course, we don't want that the healthy cell types die also. So we tested also on these 83 different healthy cell types here. This was kind of our toxicity test. And based on this analysis, we screened more than 3,000 different antimetabolites. We identified about 100 antimetabolites that could work for HCC. We looked across these, their function. Well, here's actually the reconstructed gems. You can see that there's a generally consistency, but you can see that there are actually some individual phenotypes in the different HCC patients, some parts of metabolism that are not present. And this we can also see when we compare the metabolite or the enzyme differences. And this is of course a clear illustration of these differences, but that can happen in connection with cancer. Liver cancer is, however, not a really good case to illustrate this because, as I mentioned before, liver expresses practically all enzymes and so therefore, even if certain parts falls out, it can be compensated very much by other reactions in the liver. So, but these antimetabolite identification led us then to, as I said, to begin to zero in and identify parts of metabolism that could be drug targets. And these are the 101 antimetabolites we identified. You can see that not all of them will work equally well in all the patients. So some of them seems to work in several of the patients, for example here, nicotinamide and glutamine also. And some of them would, of course, not really work as strong antimetabolites. But what we did was that we identified some, and actually 22 of these antimetabolites identified are currently already in use as anti-cancer drug targets, and 61 have earlier been proposed or shown to have strong correlation with cancer progression. And so that was a strong validation of our findings here. However, we wanted to further validate this, and one target that we identified was L-carnitine. L-carnitine carnitine is very much generally recognized as being associated with cancer development. That's why it's generally not recommended to eat so much red meat, because it has a lot of L-carnitine in it. L-carnitine is playing a very important role in bringing in a cell group into the mitochondria for beta oxidation. And if we block L-carnitine or have an antimetabolite of L-carnitine, we'll basically block this carrier transport here and prevent the cells from beta oxidation. And there's already been identified antimetabolite, as I mentioned, perhexaline. So we were looking into this, and we then benchmarked it on HepG2 cells. So this is a cell line often used for HCC cancer in our studies. And we benchmarked with sorafenib, which is currently used for treatment of HCC. And what we could see was, first, we were a little bit disappointed when we used 4 micromolar you could see there was no effect, but when we went up in dose, so probably there need to be still some chemistry development on this drug, but you could see then we could get equally well killing of the cells as we can with this current market drug. So that was some older studies we've done. More recently, what we've done is that we have taken the human model and updated it into and also taken information from various other sources. There are several other models presented and available in the literature, Recon 3D, for example, but also this IHSA model here. We incorporated information on that together with our previous model, HMR2, into what we call human 1. And we did that in a fashion such that where we use GitHub for collection, and so we continuously have a master branch, but we can actually add individuals can add genes. So several people in my lab were working on this in parallel. There's one person that is responsible for overall curation, and so this is actually a platform for anyone to engage through GitHub on curating and further improving this model here. And anyone, and of course on the way we will publish updates on the model, and anyone who's contributed here, it will actually be tracked what changes they did and who they are, and so we can even go back and ask if they want to be co-authors in future papers on this. We also, on the Metabolic Atlas website, we also made available a visualization of the models. Visualizations of these large models is extremely complex, but what we did was we both have traditional metabolic maps as well as we are currently in the phase of launching a 3D visualization unit. But more interesting maybe is that you can search for a specific enzyme and then look at all its interaction partners. That means all the metabolites that it is interacting with. And this is now also available through the the Protein Atlas. That's an extensive resource on human proteins. And there you can then enter in and actually get information on this, on the availability and then the link to metabolites. We also took human 1 and developed a so-called Gecko model. I talked about this Gecko model where we add this information about enzyme constraints. When we did that, we could really see that we could improve the simulation performance. This is illustrated over here, where we can see really the relative error in the model is significantly improved with the EC model compared with standard FBA simulations. We can also see that we did, for those who are really into the details here of flux balance analysis, that the flux variability really significantly decreased when we have this kind of model, meaning that we have less reactions that are non-determined fluxes in the network. We also then worked on taking this model to make cancer-specific models. So we actually developed models for close to 5,000 cancer patients. This was based on RNA-seq data from this. This is available through the Cancer Genome Atlas, and this was 13 different cancer types. And then we mapped what we, for example, have of specific reactions, with core reactions that are in all cancers, as well as pan, and some that are shared between. We also found some that are contextual and some that are absent in all cancers. And so this global mapping actually enabled to also begin to search for why certain drugs may not work, because they're targeting enzymes that are maybe even absent in many of the cancers. So then let me move on and talk a little bit about how we can use these models for biomarker identification. And actually this came as a little bit of serendipity, because we were starting off on a different track. We were, back several years back, we were interested to see whether metabolism of cancer cell is different from normal cells, and whether it's more similar between cancer cells compared with the tissue of origin. So we looked into RNA-seq data from 10 different tumors, and we also had data from the healthy tissues from the same subjects, and this is also data from the Cancer Genome Atlas. And when we looked into this data here, we saw very clearly here, we saw very clearly a separation here of clear cell renal carcinoma and also healthy kidneys. So this was a little bit surprising to us that we did see this very strong distinction. And so it sidetracked us actually a little bit, and to say, okay, maybe we could use this very distinct features, of kidney cancer to look for biomarkers. And the idea there is, as I mentioned before, that you have this global network. We now have a cancer network here. We overlay transcription data. It can be RNA-seq data, but it can also be proteomics data from healthy and cancer tissue. And then we find that certain pathways are upregulated or certain pathways are downregulated. Then we say, okay, so the metabolites associated with upregulation here pathway are likely to be more abundant, and metabolites associated with this one over here are likely to be reduced level, and that can then live to guide the identification of specific biomarkers we can now look for in plasma. So that was the rationale that we had. And as I said, when we did the analysis of the 10 cancers, we found that clear cell renal carcinoma, which is the most dominant form of kidney cancer, was really separating from all the other cancers. So this clearly some metabolism here that is really altered. There are many reasons for this being so different, and I'll actually not spend time on that. You can go back and look into the original paper. But one thing that was very striking was that we found a distinct deregulation of glycosaminoglycan metabolism in kidney cancer. And so this is seen here. Here's shown the network component of this. So this is the network part of this part of metabolism. You see red here are all the enzymes that were upregulated. Blue are all those that were downregulated. You see they're highly connected here. So they could really point to that there is a coordinated deregulation of this. If we overlay these enzymes that are upregulated and downregulated into a more traditional metabolic pathway map, we begin to see a pattern that it's upregulation of chondroitin sulfate metabolism and it's downregulation of heparin sulfate metabolism. And we validated this by immunohistochemistry of specific enzymes engaged here. So there is indeed also changes in protein pattern in clear cell renal carcinoma of these enzymes. So that led us then to begin to analyze the glycosaminoglycans, heparin sulfate and chondroitin sulfate in plasma and urine. And we did that in clear cell renal carcinoma patients versus healthy control. And we measure a whole panel of different chondroitin sulfates and heparin sulfates. And so actually in total 19 metabolites are measured across the board. And we use all these 19 measurement data here, then using machine learning to generate a combined score and here's shown both in plasma and urine samples individually. And so you can see here that the computer-generated score gives a clear distinction between the healthy and the cancers. And particularly when we have the combined here, we actually get an area under the curve of 1. So really excellent prediction. Should be mentioned though, this was a relatively small cohort. We had, in this case here, 34 kidney cancers versus 16 healthy. But however, we have taken this further. We've validated this in 4 following clinical trials, and the spin-out company Ellipta is currently running a pivotal trial engaging more than 500 different subjects for approval of this biomarker for clinical use. And this will typically be for recurrent of kidney cancer. The current treatment for kidney cancer is operation. You remove one of the kidneys that have the tumor in it. But there is a recurrence rate of cancer of about 50%, and you would like to monitor these people along the time in order to be able to treat them at a very early on stage if they have metastatic cancer appearing. So this is exactly as I said, it permits timely treatment of recurrence, but there are other business models that can be pursued. It can also be that you are under drug treatment and you're not responding to the current drug. You can switch then faster with this because you can have easy monitoring. It spares also unnecessary surgeries. This is maybe a harder one, but actually 20% of venous masses that are removed on operations are benign, so they don't need to be operated. But also, as I said, it allows early diagnosis, which is particularly important in connection with metastatic development. So we have, as I said, already validated this extensively. This is from a large study in collaboration with Memorial and Sloan Kettering Cancer Center of about 180 samples. Again, you see very good separation. We could actually, these subjects here were distributed into various types of cancer, localized, locally advanced, or advanced, even different stages of the cancer. And you could see that in all cases here, even with very small tumor, you can see here, even of 3 centimeters of size, the biomarker could actually identify this. And also from this study, you see a very strong prediction of it with a sensitivity of close to 96%. So does this biomarker also have prognostic value in terms of survival rate? And so here what we did, we followed a prospective cohort of 31 patients for a median of 2.6 years. And here you can see that patients with a high score have a higher mortality rate compared with a low score. So it really also has prognostic value in terms of design of, for example, treatment strategies. This was here shown for urine, the same goes for plasma. So we've also, we would then, with this success here, we then went back and actually said, okay, let's look at the transcriptional regulation also in other types of cancer. And it turns out that even though kidney cancer had this distinct metabolic phenotype, that is actually manifested due to many other reasons, whereas glycosaminoglycan metabolism seems to be generally deregulated in cancer. But it's deregulated different. So if we take one cancer, it's some genes that are up, and in other cancers, it's other genes that are up, and so on. And so, but when we looked at, for example, prostate cancer, we could also find a clear marker that led us then to identify, look for a biomarker in prostate cancer. And we, you could see here also same principle again. We measured glycosaminoglycans in urine and plasma, and you could see clear separation between healthy and cancer, as well as combined score is really strong. Interesting enough, if we use the machine learning generated composite score for prostate cancer, you see that it is specific for this, and whereas it's separate here for RCC. Whereas if you take the RCC score or machine learning algorithm for RCC, you can see you separate also here. So you really have specificity to some extent between the different type of cancers. We recently worked on expanding this also to cover protein secretion, because you would also like to be able to pick up proteins in plasma as biomarkers. And there are protein secretion is of course immensely complex in urocarial system because you have secretion through the ER, Golgi system, and eventually secretion vesicles. But the secreted proteins really play a very important role in human physiology. And they're also a very important role in cancer. You often see many more proteins being secreted, and also different proteins are being secreted. So we were interested to look into this. So we generated first a model for the human secretome. We mapped all proteins that are secreted through classical secretion. And we see that there are also some proteins that are secreted by other means, for example, exocytosis. But we see here that really a lot of those that go through the secretory system are actually just targeted to the cell membrane. They're still important because they're receptors, transporters, and so on. But about 1,500 proteins are really secreted out of the cell into the circulation system. We generated this model. I'm not going into great details about this, but we generated a model for the secretion system also. It's interesting to note that there is about 600 components of the protein secretory pathway in human cells. And so these 600 components, they actually have about 4,000 clients. Of course, not so you actually have a relatively high flux of proteins going through this pathway here, but of course not all the components of the secretory system is used for secretion of each protein here. So, for example, if you look at the pancreas secretome, you will see this has evolved, of course, to ensure proper folding of proteins that are rich in disulfide bridges, typically enzymes, but also isomerases and foldases are therefore highly expressed in pancreas because you have many enzymes that contain disulfide bridges. So this is very specific, you could say, for the pancreas secretome, that it is geared for efficient and fast secretion of a number of enzymes here. Whereas if you compare that with the brain secretome, this is really more for translocation of proteins to the membrane. The neuronal cells are characterized by having a large number of receptors in their membranes, and so therefore that secretory system has really evolved to ensure that you can get proper folding of membrane proteins in the surfaces of the neuronal cells. So we were interested to look also, as I said, to look for biomarkers. So again, we took data from the Cancer Genome Atlas, RNAseq data from a number of different cancers here across. We will look into, you can see this is just compilations of the number of data sets for tumors and normal as such. And then we looked into the different tumors. Here is a little bit like the PCA analysis I showed before. And we looked into particularly when we have cases where a liver tumor sample can be compared with a paired normal tissue sample from the same patient. And so from that, we could then run through, look at specifically at the secretome, and we could then find a number of secretome genes that are really increases above all normal tissues, because we don't want to have a biomarker for liver cancer, for example, a protein that is maybe highly upregulated in liver cancer, but then it's also secreted already by many other tissues. Then it will not be a good biomarker, of course. But here we're looking specifically at something that is highly secreted in the tumor, but not secreted by any healthy cells in general. And so there we identified a number of proteins, as you can see here, that have these roles, and these are for different cancers over here. So you can see we have some that are quite specific for certain cancers here. But you can also see that we have certain proteins here, for example, here that are characterized and associated with a number of different cancers. And you will, of course, be trying to look at those that are specific for cancers and move ahead with those. This is just a more zooming in of some of the specific proteins here. And here you can see some of them being specific. As I mentioned, we would like to have them, some can be serving as multi-cancer biomarkers, but also we would like to have specific cancer biomarkers. This is an example here of some of the specific one. So with that, let me move on and talk a little bit about, and ending by talking about using the same modeling concept to study another complex system, namely the human gut microbiome. And we have even started to talk about the human gut microbiome as being an organ. We have about 2 kilo bacteria living in the human gut. They interact with us in many different ways, both in terms of immune response, but also in turning over, of course, many of the metabolites that we are eating, but also degradation even of metabolites generated by the liver and so on. And we have a lot of dynamics. It's known that we have dynamics over age. So our gut microbiome is changing from childhood to toddler, to adult, to elderly stage. But exactly how those changes are associated with disease development, we do not know. How this is studied is that we take a microbial community sample, extract total DNA, do extensive sequencing of this, and thereby then find sequences for specific bacteria living in that ecosystem, and we can map that, for example, to known sequences. And this is just an example of this concept here. This is a study we did several years back now, where we were doing this metagenome analysis of patients, type 2 diabetic patients compared with healthy controls. And you can see here we could map many of the sequences to different bacteria. We then correlated the abundance of these bacteria here with abundance of key biomarkers related that are typically used in the clinic for type 2 diabetes. And you can see that here you have a set of bacteria that are strongly positively correlating with many of the biomarkers that we don't want to have at high level in plasma because then we have diabetes, whereas we have some that are strongly negatively correlating, and it kind of doesn't give any causal correlation here, but it gives a clear link that there's a distinct microbiome in the type 2 diabetics compared with the healthy control. We then moved on and began to build models for different bacteria living in the gut ecosystem. And then we used these models to actually describe a case where people were undergoing a dietary intervention study. And we found that people that have a reduced gut microbiota diversity, that means that they have less differences in bacteria compared with those that have a really diverse one, they have a reduced turnover of amino acids. And this leads to elevated levels of these metabolites in plasma. And elevated levels of amino acids in the plasma is normally seen as have people have, if they have that, they have increased disease risk. So, and actually what we were observing was that these people with this reduced reduced gut microbiota diversity, they were actually benefiting more from undergoing dietary intervention, so meaning on a reduced diet and particularly a reduced protein diet. And so this was a clear effect, and we could see that there was a reduced level in amino acid level in the plasma of these patients, and this was also predicted from the modeling. I'm often being asked and said, well, it's known that we have thousands of different bacteria in the gut system. Can we ever move on to model everything that is going on? And so we therefore started to look at, let's look at the real scale of the problem here. And so we were looking into abundance of species over a large different number of cohorts. And there we found that in reality, there is really a set of core species comprising about 100, a little bit more than 100 species that you can see have a relative abundance close to 1%. So actually, if we take these 113 core species, we will normally account for something like 80% of the mass of the bacteria there. So this means that, yes, there is a very large diversity, but in terms of really turnover and of metabolism and so on, it is these core metabolites, core species that are playing a key role. Whereas many of the others are maybe more playing roles in terms of immune stimulation and other factors, which can be super important, of course. But in terms of metabolism, we can probably capture the features by looking into this set of core metabolites. So we therefore started a project where we were saying, could we build a model where we had dynamic simulation of changes in the gut microbiota? So for that reason, we zoomed in on initially just 8 bacteria, and we're currently working on expanding this to a much wider cohort. So 8 bacteria. We collected kinetic information about these species here. We generated so-called reduced model using the concept of elementary flux modes. And so we have to combine with the kinetic information and the model simulation, we can actually begin to simulate the dynamics of these species. We could compare, some of these species have been studied also in the lab. And we could compare growth of these in the lab with experimental data, and then we could really see that we could get good fit of the model to these experimental data. We could also actually see that we could describe also mixtures, co-cultures, for example, of 2 bacteria as illustrated here. These are experimental data down here. These are model simulations up here. And this enabled us then to look at what is the interaction between these 8 different species here. You can see most of them negatively impact each other. They're competitors. Some of them are positively synergistic interacting with pluses here. Some are zero interacting here. But what you can see is that most are really negative interactant. And that's what is often found in ecosystem stability, that actually you have negative interaction on them, and so you have competition. That doesn't mean they cannot coexist, but because remember, this is a continuous replenishing system and quite complex, but it really impacts or demonstrates what is the key ecological driver. So we then moved on to say, okay, now we have models for the individual bacteria and how they can interact. Now let's build a model for the gastrointestinal system. And so what we did, and here we took a typical chemical engineering approach, we simply described the large colon as a multi-tank in series model. So 4 tanks in series. And actually it's because of both the lumen, as well as here the layer here close to the epithelial cells. We actually had, for each tank, we had a tank, a separation tank, so you have a stagnant layer, so to speak. And with that system, we could now simulate, and this is a quite complex model because it accounts for also water absorption, as well as absorption of metabolites into the bloodstream, as well as, of course, some element of backmixing due to peristaltic movements of the bowel, as well as we then, of course, have rectum here. And of course, it's very hard to intervene and get data on what is the microbial community in different stages of the gut system here. But with this model, we can begin to simulate this. And in order to validate the model, we took model simulations and then predicted the abundance. And this was from a cohort of Swedish babies moving from breast milk to solid food. They were monitored with a fecal sample after 0, 4, and 12 months. And here you see the abundance of the individual species over time. And these are for model simulations, and here are the abundance across the population. And you can see a model is capturing this very, very well. We validated also using another study where people underwent a ketogenic diet, and we could again also capture changes that were occurring in these changes in this smaller cohort of adults. As I said, what is of course particularly interesting is that we can begin to look into the dynamic evolution of the gut microbiota over the 12-month period. So we can see here how specific bacteria colonizes. These are the different reactors. Color indicates the abundance. And we could see here, for example, how these species here are beginning to colonize later in the 12-month period. Some are early colonizers and then actually disappear again, and so on. And we can see that throughout the time, as well as also in the different stages of the colon. We can also do that for different metabolites. That is, of course, fluctuating depending on diet intake. And but in all cases, we could see what is then the particular level of metabolites. And you can see, for example, hydrogen and butyrate are particularly produced in the lower part of the colon and also after colonization in the 12 months. So we were also engaged in a study with the Bill and Melinda Gates Foundation about whether we could begin to do these model simulations to identify what are the dietary requirements of stunted children. And there we actually did identify a clear lack of amino acid biosynthesis potential in the stunted Malawian, and it was in a study from Malawian and Bangladeshi cohorts. And therefore, they do not actually respond to provision of ready-to-use therapeutic foods, and therefore, really, they need to probably be given probiotics in order to enrich their microbiota to be able to deal with amino acid biosynthesis. So that brings me to my summary. I hope I've demonstrated to you that genome-scale models, metabolic models, can be used to identify novel drug targets for cancer treatment. They can be used together with integrative analysis of omics data to find co-regulated networks that can point to novel biomarkers. Glycosaminoglycan profiling represents a very strong systems biomarker for detection and prognosis of clear cell renal carcinoma and prostate cancer, and actually we moved on to several other cancers. And the concept of GEMs can be expanded to cover the protein secretory pathway, and this can begin to lead to identification of protein biomarkers. But GEM can also be used to get insight into the dynamics of the human gut microbiota. And with that, let me thank all my excellent students and postdocs and collaborators over the years, and for your attention. Thank you very much.
Are there any questions, comments from students or professors? So right now we are waiting for some questions from chatting room. Okay, Jans?
Yeah?
Can you see the Kayong Lee's question?
I can see the chat. Yeah, I can. Yeah.
Yeah.
Okay. Okay, so the first question goes about how do we go for human GEMs in terms of flux simulation, and this is a very good question. So this has been challenging us quite extensively. We have in a few cases been doing flux simulations for example, for drug identification, as I illustrated here for liver cancer. We did a comparison actually also on what was the predictive strength in terms of knocking down enzymes compared with sRNA screen for two different cancers, and we used cell lines to validate that. And it turns out that the model predictions were not really very strong. So there would be a number of false positives when you do traditional FBA. I think, though, that the modeling are often given this, overview and points of metabolism that can be interesting to look at and pursue further. And particularly also after we have generated the EC-GEM, the model that has kinetic information, we have a much improved model, and it still, of course, needs to stand how that will compare. I didn't talk too much about it here, but actually on the slide, there was also, we did a comparison of how that performs in terms of predicting essential genes in cancer cell lines, and it performs much better than the traditional FBA simulation. So I think there's some progress, and we'll see more and more progress. But the challenge is really that we have very poor understanding of the constraints. And when I talk about constraints, that is the so-called exchange fluxes. When we are looking at microorganisms, we know exactly how much glucose is coming in and so on. We don't know that for human cells in the body. And so therefore, we have to have many more loose exchange fluxes, and that causes problems. Of course, that combined also with the larger scale of the model. The next question is about, do you think we should use single-cell RNA-seq rather than bulk RNA-seq to reconstruct tissue cell context-specific GEMs? And this is also a very good question. We are actually working quite extensively on using single-cell RNA-seq. The challenge with single-cell RNA-seq is still that they're extremely noisy. So it's hard to get really good high-quality data for this. But the idea, of course, is that when we use a tissue sample, it is a mixture of cells in that, and we are very interested in actually beginning to build whole tissue models based on saying this is the metabolism, for example, of the different cell types that are in that tissue, and there we would have to have single-cell RNA-seq data, at least initially for that. Maybe in the future we can have it from bulk RNA and then have quantitative information about the relative abundance of different cell types. Then we can begin to extrapolate from that. I hope that answered the two questions. Very, very good.
Yeah, thanks so much. So is there any comments and questions? Hello, you have questions?
Yeah, I have a short question. First, thank you for the great talk. This is a very soft question. Just curious how human genes are perceived by medical doctors in your area. Like, were they kind of skeptical when you first heard about the possible application of human genes for collaboration work? But then, are they like convinced right now, or is it well received in the medical community? I'm just kind of curious about that atmosphere.
Yeah, that's a very good question. So, this was a very positive surprise for me, I must say. Coming from the traditional field of biotechnology, microbial fermentation, and so on, and going into the medical research, I was very surprised how open medical doctors were, looking into new alternative ways of doing things, because they really just care about getting insight so they can cure diseases. And so they, in many cases, and that of course, I've also only interacted with a subset of population of medical doctors, but those we have interacted with, which probably count to more than 20 in many different countries at least, have been very open-minded on this, and really believe also in the power of computer models to be able to develop either better drugs, biomarkers, or also even help them in the clinic for diagnosis. I can maybe add a comment to it, but maybe one thing, what of course was challenging for us in the beginning, was to get a thorough understanding of the language used, but also get a real understanding about what are the challenges, what are the medical challenges for the doctors in order to move forward. And so that took some time to get to that. Of course, eventually you get to learn the terms, the naming, and so on. So the language gets fixed, but it was a longer process to understand. And so therefore, I'm very keen always when we embark on a collaboration to ask what would they like to get out of this? What objective would they see from this joint study?
Mm-hmm. So then, what would be more limiting for the wider application of human gen? Would it be the further improvement of a model, or would it be the more availability of patient-specific omics data?
I would say we are probably not that limited by data anymore. There are even a number of public databases and so on, where it's directly answering, it's probably still an emphasis on improving the models. But I think though that where we have a lot of data from is cancer, and particularly through the Cancer Genome Atlas, but also other sources, other publications and databases and so on. Where we also have a lot of data is for many of the chronological diseases, cardiovascular disease, obesity, and so on, type 2 diabetes. However, those diseases are often associated with many comorbidities, as I said. So it has been very hard. We are engaged now in a study on people undergoing bariatric surgery, where we have multi-omics data from these individuals that you could normally not get, but you can get them because you're operating them at the same time. And that begins to show some very interesting findings, I would say, in terms of also the link between the different diseases and so on. And there is a question in Korean. I'm sorry, I cannot read that.
That is not question.
Okay.
That is not question. Jens, I have a very crude question, very naive question. You know what? In the drugstore, we have probiotics, right? Pill.
Yeah.
Normally, we take it for diary purpose. So if you have your model used for formulating this kind of a bioprotic pills. Is it possible? So, for example, you take out some data from one person, and then you just customize that guy's bacteria in the gut, and then you can customize the pill for bioprotic, bio.
Yeah,
Propensity.
exactly. No, exactly. I understand your point.
Yeah. For that specific human being, is it possible?
I think it's possible. To answer very simple. That was the rationale and the objective for this study. We want to be able to go in that direction. But where it is complicated is that we only have 8 species there right now. We probably need to expand that to at least 20, 30 in order to get a more broader coverage. I don't think we need to necessarily go to the 100 I talked about in the core microbiome. Mm-hmm. Because many of them also have similar type of metabolism and so on. I didn't talk too much about that, but if we maybe go to 20 and 25, we could have a pretty good representation. Then we could simulate what would be the impact of, for example, taking Lactobacillus reuteri or another probiotic that you can buy. Most of these don't colonize as such. That's why you also need, and that's of course nice for the producer because they need to you need to take it all the time. But then one could begin to simulate the residence time and how that impacts and the gut microbiota. We did a small scale study, I can say, with in collaboration with Danone some years ago, where we were looking into this. There we didn't have the refined models as we have now, but there we were looking into this, particularly some of the bacteria that they have in their Activia product.
So another naive question is, so what is the difference between yogurt and probiotics functions? I mean, people sometimes confuse the role between yogurt and the probiotics. So can you give us some comments on the functionality of yogurt and the probiotics?
Yeah, of course. I mean, so let's not forget that if you go to the drugstore and buy probiotics pills, many of them have never been clinical validated, right? So these are, it's not like vitamins that you buy. If you're low on vitamin D, you take vitamin D pills. This has not been validated to the same extent. So therefore, there's a lot of uncertainty, I would say, associated with this, and much of it is based on belief more than actually scientific evidence. And so we need to get scientific evidence behind the role of many probiotics. They may play very important roles, but what I think is even more important is what I would call second generation probiotics, actually specific bacteria isolated from the human gut, purified, made into second generation probiotics with the objective of redirecting the gut microbiota, actually colonizing and redirecting it permanently. That is more, you could say, second generation probiotics as a drug rather than the other ones that are transiently passing through the gut system.
I see. Thank you. So you can see some questions popped up.
Yeah. So there was some question about finding target biomarkers for the COVID-19 pandemic, for enzyme targets for blocking or attenuating the infection. We have not taken any efforts into this. I think it's a good idea. I think though, and I said I'm pretty sure that there is a metabolic fingerprint of phenotype associated with COVID-19 infection. But whether the solution for treatment is metabolic, I am maybe skeptical. So this, I think, is really other parts. Maybe not surprisingly, I mean, some targeting nucleotide metabolism, for example, would of course have an impact because the virus is replicating and it needs nucleotides en masse. So that has also been, I think, some studies been shown. But I would look into other parts of metabolism or physiology, I would say, or cell function. And we have, as I said, not taken any studies into this. I know some people have. Okay, so the next question is in the sense of responding to outside. I think your Genomescale models have limitation for handling those kind of. Have you been thought about integrating a gem along with regulatory network? Very good question. Also, that regulation. Of course, we are not covering that. We have done that in yeast, where we've added the regulatory network on top of it, on top of a gem, and then to simulate. The key challenge, though, we are facing there is, and that's why we are looking into metabolism so extensively, is that is an extremely curated part of cellular function. As soon as we go to regulatory networks, there are many more uncertainties. And there are also uncertainties in the sense of kinetic information. So we may know that this kinase may activate these two enzymes here, but we don't know that if it gets, this enzyme, for example, gets phosphorylated, how is its activity changing? We don't even know if it's going up or down, and even if it is going up or down, is it going up by 30% or 50% and so on? And all that information becomes then suddenly important if we're going to have value for adding the regulatory networks on it. So yes, we are neglecting all this, and so we are taking, you could say, just this snapshot view. But if the objective is not really to quantify all the processes in the cell, but actually using the model as a scaffold for getting some sort of enrichment of information that we have from the omics data, then I think one is better off by not having the regulatory structures in there. With that said, I think it's definitely an interesting avenue of research, because it is really only through beginning to add these regulatory networks or models into a genome-scale metabolic model, for example, that one can begin to validate many of the hypotheses that are out there about the structure of these regulatory mechanisms. So I hope that answered that question. Then there is some question here about yes, the first question is about human metabolism. Do you think tissue types can be distinguished by intercellular flux distribution? And the answer to that is yes. Not all, some tissues are very similar type of metabolism. We see this specifically in terms of which enzymes, as I showed, which pathways are expressed. But even in terms of if we do simulation and calculate fluxes, we also see very clear separation. We actually did that predominantly for cancer, but we will see it also for different human tissues. So the calculated fluxes is actually something that I'm quite keen on moving forward with, because that is, and it was another question also related to how good are the fluxes predicted. And I think with the EC model, we begin to get good value of the fluxes, and the fluxes are actually extreme phenotypic discriminators, I would say, because they are really a consequence of many different layers. A gene is expressed, a protein is there, this protein, and all these proteins are there. So now we can have flux through this pathway. And how big is that flux is depending on the environment and so on. So that information is high-level information that I think could be particularly interesting to use for example, machine learning, to then find specific features. The second question is, I'm wondering about which metabolic flux simulation algorithm is commonly used for studying human metabolism of normal tissues, not a cancer, in your research group. Do you think flux balance analysis is still acceptable enough for normal tissue specific? Yeah, that's a very good question, because what is the objective function in these cases? And there we have worked on trying to specify some objective functions. We have not pursued this, I would say, extensively, because we have predominantly in modeling pursued cancer, but one would have to specify some sort of objective function. And here again, the EC model could potentially help, because one could use a criteria, for example, on minimizing total flux set, as has been done in some one of the algorithms used for flux estimation. So I hope that answered the question also. A lot of very, very good questions. Thank you very much.
Yeah, that's great. So any further question or comments? If not, let me stop here. And then I should say thanks so much for your fabulous talk. And then if any student or faculty members has questions, let us summarize a little bit and send it to you by email. How about that?
Thank you very much. Thank you.
Thank you so much, and then have a great fall semester there.
Thank you. Thank you.
So thank you for participating in this 10th annual distinguished lectureship. Thank you.
Thank you very much for inviting me. Thank you. Bye-bye. Bye-bye.